IB Bio Topic 11, Animal Physiology Part 2, Antibody Production and Vaccination B, has its focus on vaccinations and monoclonal antibody production and use. The essential idea is immunity is based on recognition of self and destruction of foreign material. Now, I've chosen this slide to emphasize an important point within this unit. Every organism has unique molecules on the surface of its cells, as can be seen in this image of a cell membrane. Molecules projecting from cell membranes are unique to each species. These molecules allow our body to distinguish self from non-self. These species-specific molecules are critical to the defense against specific pathogens. Here is an outline of all the available movies in Topic 11, a topic for higher level students only. Use this outline to find the movie you need for review purposes. This movie is focused here. I had this slide in Part 1, Immune A, and I've started this movie with this slide to refresh your memory. Molecules on the surfaces of our red blood cells distinguish us as either type A, B, O, or AB. The molecules on the surfaces of our red blood cells are potential antigens if blood is donated to a person of a different blood type. And each blood group produces specific antibodies in response to the antigens. Thus, we must be very careful about blood transfusions, because if type B blood is donated to a type A person, the type A antibodies will bind the type B molecules on the red blood cells of a person type B, causing clumping of the blood in the recipient, and this could potentially lead to death. I've come back to this slide repeatedly because it's very good to study as a way to understand both the big and small picture of the specific defense system. An antigen arrives on the surface of a pathogen. The antigen is a molecule on the surface of a pathogen recognized by B cells, macrophages, and T cells as foreign. B cells recognize the presence of the antigen and are stimulated to divide into many B cells, a clone of identical B cells, some of which immediately make antibodies that will bind the antigen in the bloodstream. And some B cells remain alive within the body as memory cells, ready for a second encounter with the pathogen. Immunity depends on the persistence of memory cells. Now, most of you have had chickenpox and are now immune from another infection of chickenpox. Most of you have had a vaccination for measles, mumps, rubella, so that you will not experience an infection from these pathogens. Your immunity from these pathogens comes from memory cells that remain from a first encounter with the pathogen. Here's the next IB syllabus statement. Explain the principle of vaccination. The fundamental principle of vaccination is something you already understand if you understand the nature of memory cells following exposure to a pathogen. Vaccination is done as a part of healthcare, first injecting or consuming small amounts of the pathogen with the antigen, and then this causes clonal selection, including memory cells. This is a primary response. The memory cells remain behind or stand ready to trigger a second rapid response if the pathogen presents itself again. And the rapidity of the second response prevents recurrence of the disease. This is how vaccination is responsible for immunity. Remember from the last movie the benefit of having memory cells in your body. Following a first exposure to antigen A, there's production of antibodies, and then here we have memory cells. And then a second exposure occurs, and the number of antibodies produced by those memory cells residing in the body increases dramatically over a relatively short period of time. And in this way, you are immune to the pathogen. Here's how a vaccine is made. A piece of DNA from the herpes virus is removed, and the piece of DNA is inserted into the DNA of the harmless cowpox virus. So the DNA with the herpes DNA is inserted into the harmless cowpox virus, and this is the vaccine. The vaccine is then injected into the body. Now, the body is not receiving a virulent herpes virus, so the patient will not get sick. The patient is receiving some herpes DNA that will make antigens. Upon injection of the vaccine, B cells of the patient are stimulated by the presence of the antigens, and the B cells clone. Then memory cells remain in the body, so if the body is ever exposed to herpes, the body will not get sick. The body has immunity as a result of the vaccination that stimulates the production of B cells, including memory cells. So active immunity is the active role of lymphocytes, B and T memory cells, in the defense against an antigen, a pathogen, to which the host has already been exposed through either previous direct exposure, in other words, an infection, or from a vaccine. Again, it is the active role of the B and T memory cells in providing immunity based on a previous direct exposure or a vaccine. Now, are there benefits and dangers of vaccination? Yes, and you need to be able to discuss the benefits and dangers of vaccination. So the advantages of vaccinations are that they protect you for many years against the pathogen. In other words, the response to the second exposure is fast, so fast you may not even realize a second exposure has occurred. A second advantage, total elimination of the disease, for example, smallpox. 3, prevention of pandemics or epidemics, decreased healthcare costs, and prevention of harmful side effects from the disease. The dangers of vaccination could be that vaccines are virulent, cause illness, possibly death. The vaccine could be an allergen. Some vaccines are preserved with mercury compounds, and the mercury exposure could be dangerous. Vaccinations might overwhelm the immune system. Is autism associated with the mumps, measles, rubella vaccine? And vaccinations might give a false sense of security against dangerous pathogens to which you might be exposed. And if you subscribe to the belief that vaccinations are terrible for children, you run the risk of having your children get sick from diseases that most people are barely aware of. This graph shows the rate of measles and measles vaccination coverage in England and Wales. In fact, vaccines have been hugely effective in reducing disease. With the arrival of the measles vaccine, 1968, and coverage across the UK increasing, the number of measles cases in the UK declined dramatically. The measles vaccine had the same impact on measles in the United States. Vaccination skeptics have argued that better sanitation, not vaccines, were responsible for the decreased death due to these pathogens. Now, if sanitation were responsible for getting rid of measles and other diseases, you would expect all diseases to decline at roughly the same time. However, not all diseases decline at the same time. The decline in specific diseases corresponds to the introduction of the vaccine. While good sanitation is responsible for the reduction of many diseases, cholera, dysentery, typhoid, vaccines are responsible for the reduction of diseases like smallpox, polio, and measles. As a note, the small increases in measles cases seen here, here, and here result from people choosing not to vaccinate. History's largest clinical trial, which involved over 1 million children, was conducted on the polio vaccine in the U.S. in 1954. Notice how quickly the number of polio cases declined after 1954. Now, this doesn't conclusively demonstrate that the vaccine stopped the disease, because correlation doesn't always mean causation. However, in this trial, children given the vaccine were significantly less likely to get polio than those who were given an inert injection called a placebo. This fact, combined with the graph, demonstrates fairly conclusively that the vaccine for polio is what reduced polio in the U.S. There has always been skepticism about the use of vaccines. Being injected with a pathogen, even a weakened or dead pathogen, could make anyone a bit nervous about the consequences. In Japan, rates of whooping cough were low when vaccines were given, but when the vaccination programs were dropped, the incidence of whooping cough increased. Until the vaccines were reinstated, then the incidence of whooping cough declined again. Tetanus, a disease caused by the bacterium Clostridium tetani, also declined as vaccination programs were introduced in Australia. But skeptics persist, and some suggest that the decline in the incidence of measles preceded the introduction of the vaccine. In this graph, the authors suggest that the decline in measles incidence, not death, started before people used vaccines in large numbers. The authors note the decline here and through here, in addition to the fact that only 33% of the people were taking the vaccine. The same authors suggest that measles death rate declined well before the 1968 introduction of the vaccine. Interestingly, a 2011 study out of Germany compared the incidence of various illnesses seen here along the x-axis as a function of being vaccinated in blue or not vaccinated in red. The study suggested that vaccinated children have at least 2 to 5 times more diseases and disorders than unvaccinated children. The smallpox virus, seen here, was the first infectious disease of humans to have been eradicated by vaccination. The smallpox virus is a filamentous RNA virus. The World Health Organization initiated the campaign for the global eradication of smallpox in 1967. The campaign was deemed a success in 1977, only 10 years later. This graph shows the number of smallpox deaths across the U.S. in the early 1900s. While compulsory vaccination varied state to state across the U.S. in the late 1800s, a legal case in 1905 set the stage for compulsory smallpox vaccination. Once compulsory smallpox vaccination was enforced by inspectors, the deaths due to smallpox in England dropped dramatically. So here are 2 relevant IB syllabus statements. Viruses lack a metabolism and cannot therefore be treated with antibiotics. Vaccines are a successful approach toward eradicating diseases caused by viruses. Secondly, the smallpox virus was the first infectious disease of humans to have been eradicated by vaccination. Now, viruses such as HIV, smallpox, influenza, rabies, dengue fever are small particles. They require a host to reproduce. They're not made of cells. They're non-living. They lack a metabolism, so that antibiotics are ineffective. In this micrograph, we can see a viral particle inserting its DNA into a host cell. Antibiotics are not effective against viruses because antibiotics block specific bacterial metabolic pathways. Viruses lack a metabolism, and viruses used host cell metabolic pathways. Vaccines, using dead or weakened parts of pathogens, serve to overcome the viral pathogens that do not respond to antibiotics. As a last note on the topic of vaccines for humans, vaccines are often produced using the immune responses of other animals. Animals are injected with the pathogen, and the antibodies from the blood of the other animal are harvested and injected into people. Now, this is known as passive immunity because B and T cells are not activated in the human. There are no memory cells when nothing more than antibodies have been injected as the vaccine. Now, passive immunity involves injected antibodies produced and collected from another organism, or antibodies produced by a mother and passed via the placenta to the fetus, or antibodies in mother's milk. Now, the last topic of this movie focuses on monoclonal antibodies. Antibodies are large, Y-shaped proteins, seen here, produced by B lymphocytes. Antibodies bind to foreign molecules, antigens, seen here, bacteria and viruses. In this image, we can see antibodies in red binding to molecules on the surface of a virus. Monoclonal antibodies arise from a single clone of B cells that produce a single specific antibody molecule. As a result of the specificity of the antibody and the large quantity of them arising from the clone of B cells, monoclonal antibodies have various applications in biotechnology. A B cell of the immune system is stimulated by the presence of a foreign molecule, the antigen. We can see this here. The B cell responds to the presence of the antigen by synthesizing and secreting antibodies, seen here. Now, the antibodies specific to the antigen can be harvested and used in biotechnology. Stay tuned. The technique used to produce monoclonal antibodies is shown here. The antigen of interest, for example the HCG hormone, or say a blood enzyme, is injected into the mouse. Remember, B cells specific for the antigen make antibodies. The B cells are withdrawn from the mouse and hybridized to tumor cells to produce a hybridoma, which is a long-lived cancerous cell that divides and produces a clone of cells that make monoclonal antibodies. Now, the monoclonal antibodies are harvested from the hybridomas, and the antibodies can be used to diagnose pregnancy if the original antigen was HCG, or the monoclonal antibodies could be used to diagnose the occurrence of the enzyme in the blood of someone if the original antigen was that enzyme. Here's the IB syllabus statement of relevance. Describe the production of monoclonal antibodies and their use in diagnosis and in treatment. The description should include fusion of tumor and B cells to produce a hybridoma and hybridoma production of antibodies. A molecule of interest, called the antigen, is injected into a mouse. B cells specific to the antigen are stimulated and proliferate a single clone of B cells. The B cells are collected from the mouse and fused with a tumor cell that provides longevity, the hybridoma. The fused cells are selected from a special medium and they produce the antibody of interest, and a lot of antibody is harvested. The antibody is used in diagnosis and treatment. Stay tuned. Here's one final image of the technique. Please spend some time studying the process. Remember, the fusion of the tumor cells to the B cells harvested from the mouse provides a long-lived quality to the hybridoma so that the monoclonal antibody production continues over time. We'll look more closely at the uses of monoclonal antibodies in the slides to come. The use of monoclonal antibodies in diagnosis includes the detection of HCG in home pregnancy kits, the detection of HIV antibodies to determine if HIV infection has occurred, and the detection of an isoenzyme in the blood from a suspected heart attack. Stay tuned for the use of monoclonal antibodies in treatment. Monoclonal antibodies are used to diagnose pregnancy by the detection of HCG in urine. Monoclonal antibodies are the functional unit of home pregnancy kits. So if a woman is wondering whether she's pregnant, she would take a urine sample, and assuming that it has HCG in it as a result of her pregnancy, and she applies it to the home pregnancy kit stick. The HCG binds to free anti-HCG antibodies, and you can see those here. Now on the stick there are fixed anti-HCG antibodies as well, and they bind the free or mobile anti-HCG antibodies, and this creates a color change, an antibody sandwich, which creates color change. Now, there is a control region further up the stick, and this ensures proper functioning of the antibodies, and antibody arrival and binding causes color change. And 2 color bands indicates pregnancy. In the same way, monoclonal antibodies are used to diagnose HIV infection. Spend some time studying this slide. Here's the IB syllabus statement again, and here are examples of monoclonal antibody use in diagnosis. You must speak about the detection of HCG in pregnancy test kits, as well as the detection of antibodies against HIV in the bloodstream of someone who's possibly infected, or the presence of the cardiac isoenzyme in the blood of suspected cases of heart attack. The use of monoclonal antibodies in treatment includes antibodies with an attached drug could target cancer cells, or determining tissue compatibility before transplants, or emergency treatment of rabies. Monoclonal antibodies can deliver drugs to cancerous cells as a mechanism to treat cancer. In this image, you can see the monoclonal antibody delivering a drug to a cell. Monoclonal antibodies could be used in the treatment of rabies. The antibodies would bind to the rabies virus, immobilizing it. And that brings us to the end of IB Bio, Topic 11, Animal Physiology, Part 2, Immune System B.